Hi, everyone. I'm Shannon Weiman, the Scientific Communications Manager at Keystone Symposia, and I'll be hosting this Meet the Editors Roundtable today. I have with me editors from Cell Press, EMBO, JEM, Nature, the Company of Biologists, PLOS, and Frontiers Journals to discuss the state of the field, exciting new advances in single-cell analysis, tools and applications, as well as tips on how to publish in these top-tier journals. Before we get started today, I'll go through some instructions on how we'll be running the session. First, please keep your camera and microphone off during this first portion of the event. We'll start with a 30-minute discussion with our panel and then open up for audience questions. You may submit your questions at any time into the Zoom chat. During the audience Q&A, I will select questions from the Zoom chat and ask the person who submitted the question to turn on their video and microphone to ask the speakers your question live. After asking your question, please turn off your microphone once again. And please do leave the Zoom chat clear for submitting questions. All right, I think now we are ready to welcome our editors. If our editors could please join me on screen. Wonderful. So we'll get started today by having our editors give a quick introduction to themselves and their journal. We will start with Maria. Would you like to go first?
Hi, my name is Maria Polychronidou. I'm a senior editor at Molecular Systems Biology, which is an open access journal published by EMBO Press. And our journal, well, as the name indicates, publishes really everything systems biology related, including, of course, single cell biology work. And we also have a method section where we publish new methodologies with applications to systems biology. And these methodologies can be both experimental or computational or a combination of both.
Wonderful. Next, we'll have Lucia.
Hi, Shannon. Thank you very much for the invitation. So my name is Lucia Morgado Palacin, and I am scientific editor at Journal of Cellular Biology. So our journal belongs to Rockefeller University Press and is a hybrid journal. So that means that we evaluate the work in-house professional editors together with an academic editorial board. And as the name of the journal, we are interested in the cell biology works that answers fundamental questions of cell biology processes. And we also are interested in the new methods and also new data sets. We have a tools format for that. I can put the link later in the chat if any of the people in the session is interested in have a look at it.
Great, yeah, that'd be great if you can add it to the Zoom chat for everyone. One note to the audience about the Zoom chat, you will want to copy and paste any of those links because when this meeting ends, you won't have access to that Zoom chat, so do make note of that. Our next editor, Katherine, would you like to introduce yourself?
Sure, so thanks very much. I'm Katherine Brown. I'm the executive editor of Development, which is published by the Company of Biologists. So we're a small, not-for-profit charitable publisher based in the UK. And so Development, again, as the name suggests, is interested in developmental biology, but we also have a strong focus on stem cell biology. And we've really sort of seen the power of single cell techniques to advance those fields over the last few years. And so we're interested in both research papers and also techniques and resources papers in the single cell field as they apply to either developmental or stem cell biology or in fact regenerative biology as well.
Fantastic. Sheba.
Hi everyone, my name is Sheba Agarwal. I work for Cell Press for a journal called iScience. iScience is a multidisciplinary journal. We cover pretty much everything that is in interdisciplinary and on the interfaces of different fields between physical sciences, life sciences, and earth sciences. We're also open to social sciences as well as these days also medical sciences. We launched in 2018, so we're a relatively new journal from, like I said, Cell Press. And since then, we have published over 2,000 papers in these, very diverse sort of different fields within life sciences. And I'm happy to answer any questions. I will also put in the aims and scope of our journal in the chat a little bit later on. Thank you.
Wonderful. And Ines?
Hi, my name is Ines Alvarez Garcia. So I work as a senior editor at PLOS Biology. And as you all know, probably PLOS Biology is a nonprofit open access journal, and we are interested in all biology. but we publish different types of papers from research articles to methods and resources papers. So we will welcome all your papers on the exciting new methods that you have developed using single-cell biology. And I also want to mention that we are very interested in reproducibility, and we have what we call our scooping policy, in which we consider papers that have been scooped within a 6 months window. So I can encourage you to read a blog that we have called The Importance of Being Second. And I can also drop the link in the chat later.
That'd be great, thank you. And last but not least, Allison.
Hi everyone, my name is Allison Doerr and I'm the Chief Editor of Nature Methods. Chief Editor means that I run a team of professional editors, so we make the first and final decisions on all papers, of course, sending them to peer review as well, but we are responsible for deciding what we publish. We have just recently become, I guess you would call us a hybrid journal. Transformative journal is the terminology we want to use. So we offer subscription-based publishing, but we now also offer an open access option. And we really publish methods, software tools, comparative analyses, resources for across the gamut of basic life sciences research. And I just wanted to say, this meeting is particularly exciting for me because single-cell methods has been a major area of interest for us for a long time, not just in the transcriptomics, genomics field, but across multiple different fields.
Great, and that actually is a great segue to kick off our discussion. I'd like to ask each of you, what are the most exciting advances you've heard so far in the meeting or are looking forward to hearing about later today and tomorrow? So whoever wants to jump in first, go ahead. Allison, do you want to take the lead on that?
Sure. Yeah, I think this is a really nice meeting in that it brings together people who are both methods developers and more on the biological side. So, me personally, I'm very excited to see the methods, new computational tools being presented in talks, and really things that are, not just more practical, but are really pushing the boundaries in terms of what it's possible to learn about biology. So, some particular areas of interest for us have been spatial methods. We just highlighted spatially resolved transcriptomics as our method of the year in our January issue. Multimodal omics methods, I think, are also really interesting. Trying to squeeze as much information as possible out of, one tiny little cell. It's just so amazing what people are doing these days. But, I've also enjoyed seeing the biology talks, and it feels personally rewarding when I see, oh, this group is using a method that we published. It's actually making an impact in the community. That's great to see.
Great, yeah, and I think that's what's really exciting about this meeting is we get these technical advances that are just pretty mind-boggling to me, and then yeah, you get to see the vast amount of information and insight that can provide. What about any of the rest of our editors?
Well, I personally, so this is the first, oh, I'm sorry, Maria.
No,
Sheba, go ahead, and then we'll go with Maria.
Yeah, all right, sorry. No, well, what I was going to say was I personally really, really enjoy the diversity in which all of these single-cell methods can be applied in so many different systems and so many different settings. And also the fact is that, systems, organs, whatever, they are not homogeneous. And this is something that, as a traditional cell biologist, this is something that we couldn't do when I was doing my PhD. And now I'm showing my age, obviously. But now the fact that you can actually look at single cells and look at how heterogeneous a population is in a sense of, what exactly single cells are doing to contribute to a certain phenotype. I find that amazing. And maybe, I'm really looking forward to see how far we can actually go for this, probably even to look at, effective therapies when things actually go wrong, just keeping in mind this heterogeneity instead of thinking, okay, everything is pretty much homogeneous in one particular place. So I find that really exciting personally.
Yeah, definitely. Maria, you had something to add.
Yeah, I mean, I agree with what was just said. I think the combination of biology and technology is very powerful, and that's a reason why I personally was very much looking forward to this meeting. in addition, of course, to the fact that single cell biology aligns well with the scope of our journal. And I think single cell biology is very powerful for analyzing both fundamental biological processes like cell differentiation, development, cell cycle, but also disease. And I think it is really nice that the meeting combines both these aspects. So I'm really looking forward to the dynamics session tomorrow, and also the computational approaches session later on, just to see what kind of new computational methods are around for interpreting and analyzing data in new ways and deriving new insights. And yeah, I cannot single out anything specific that I heard so far, but yeah, it has been a very interesting meeting.
Yeah, that's true. And that's interesting because the methods provide so much data. Then how do we manage that, and how do we make conclusions? So yeah, I think all those pieces have to come together. Lucia, did you have, sorry, Catherine, go ahead.
Oh, sorry, Lucia. So I think for me, I was at this meeting 2 years ago in person in Keystone, which was really nice. And I think what for me is really great is just how fast this field is moving. And from sort of my perspective scientifically, as a sort of developmental biologist, I think the things that are really important that are really coming are both the spatiotemporal analysis that we can do now. So in terms of understanding lineage relationships at the single cell level, and also looking at not only is there lots of heterogeneity, but how is that heterogeneity organized in space and in time? And for me, I think that's sort of what's really exciting and where the field has really sort of accelerated over the last couple of years.
I completely agree with Catherine. So it's my first meeting specifically in single cell biology, and I was blown away yesterday by all the new technology that is available at the moment, and how the field has really matured from producing a huge amount of data that was very difficult to analyze to really be very clever, producing ways to look at the data in very specific ways to tackle disease and development, etc. So it's a very promising for the future, and very powerful to see what is going to come from this field. So it's very exciting.
Yeah.
I just wanted to add, for me, it's also my first single cell meeting, as in the case of Ines. And I was really amazed by how the field has combined different single cell technologies in order to provide like more clear picture of this heterogeneity that Sheba was before mentioning, and in order to infer like these cellular states that they might be like very important in the tissue architecture, and also in the, if we want like to predict how our tissue is going to behave in response to certain treatments or perturbations. So for sure, this is going to have like a lot of implications for better therapeutical strategies. And that's amazing how all these techniques are combined, and also how these advances provide like more resolution. So I'm pretty sure that we are going like to see in the next talks like very high and very exciting the advances.
That's true, and that's a really good point. Not only are we making huge leaps in the technologies, but combining all those technologies as well. And yeah, it's just really incredible the impact that can have. So our next question for discussion is, what kinds of stories then is your journal looking for at this meeting? What are you looking to publish? Do you have any special issues or anything of relevance? I know our audience probably is excited to contact you and see if they can publish their work in your journal. So what types of research are you looking to highlight in upcoming issues? Maybe Ines, do you want to go first on this one?
So we are really broad, a really broad journal, as I mentioned before, and we publish methods. So that would be one of the avenues that would allow you to publish in PLOS Biology, for example. What was I going to say? Sorry, I had something in my mind.
I also wanted to add, whether you're interested more in the method advancement or the application of a method to study a disease or something like
both.
that, or both?
Both. So
okay.
We are so broad that we would be interested in both. But what I wanted to add is that because we are only an online journal, we don't have any restriction in size of papers. So you can just send us any size of paper that you want to. So sometimes, actually, for methods and resources papers, that helps a lot, and is one of the things that the people appreciate.
Great.
So keep that in mind.
Who would like to go next? Katherine?
I can go.
Oh, sure. Sorry. Sheba, go ahead.
I keep interrupting people. I apologize for this. So iScience, we're a very multidisciplinary journal, and I forgot to mention that we're also open access. And the thing is that single cell technology is literally the crossroads of all of these wonderful different disciplines. We are definitely focused both on technical papers as well as like methods papers, as well as the applications into biology. The thing is about us, I think that what makes us unique is that because we are so multidisciplinary, we are not asking for just technical papers or just biological papers. We can have papers that combine both of them. And we actually like those papers because then it kind of makes a sort of a complete story. And what we do is, in peer review, we really make sure that all aspects of your paper is looked at. So, for example, I would have a sort of like a modeling person, for example, or a technical person to look at the technical bits, and an actual biologist to look at the biology of the paper. And so we do have an all-rounded sort of peer review process that I think all interdisciplinary journals can actually already do as well. But I think that is something that is quite special that we all do. And I invite everybody out there listening, please, contact us if you have any kind of any inquiries or any questions. We are always open to pre-submission inquiries, and we'll get back to you, very quickly on that.
Fantastic. Catherine, did you want to jump in?
Oh, yeah, I can jump in. So I've sort of, as I've already said, we are interested in both the biology and the technical side of things as they apply, specifically within the developmental biology and stem cell fields. We do have a technique section. We do also consider computational papers that don't necessarily have experimental data in them. And actually, we run with academic editors, and we've recently appointed academic editors to cover some of these newer fields. So for computational field and for the single cell field, Sam Morris, who's going to be speaking, I think, tomorrow, joined our team recently. And so if you're sort of interested, you can either reach out to me or you can reach out to Sam to talk about your papers. And I think the other thing that I would say is that we have a pretty active review section of the journal, and that's something that I'm very involved in. And we are always happy to look at people's proposals for review articles. And so if that's something that you're interested in sort of finding out more about or in talking to me about, then you're more than welcome to get in touch about that as well. Oh, and I guess I should also say we have a couple of special issues coming up that may be of interest to people. So we're about to close submissions for an issue on imaging, and specifically looking at imaging technologies in developmental and stem cell biology. And then we've just opened a call for papers for a special issue on the influence of the immune system in development and regeneration as well.
Fantastic. Maria, would you like to jump in?
Yeah, so as I mentioned before, we also, as we heard also from other journals, we are also a very diverse journal, and we publish both studies emphasizing new biology, but also studies presenting new methodology. Our method section is also quite diverse. I will include in the chat below a single cell biology collection that we assembled with recently published papers, so that you can get an idea of the kind of studies that we publish. Yeah, they are diverse. They cover different aspects of single cell biology, from single cell omics types of studies to quantitative single cell biology, dynamics analysis in different systems. The same applies to the methods. They're also quite diverse. So we publish computational methods that are more tailored for data analysis and integration. We also publish imaging-based methods, screening platforms, anything that can have applications to systems biology. And especially for methods, because we think that it's very important to do, we try to do our best to make sure that the methods we publish can be are published in a format that makes them widely accessible and likely to be widely used. So we have also, since a couple of years, introduced a new materials and methods format for the method papers, where we allow the authors, if they wish to do that, to include their protocols in the method section, so that someone who reads their paper can really have access to all the details, all the methodological and protocol details that they need to know if they want to adopt the method in their own lab. And we are getting very good feedback regarding that format.
All right, great. Allison.
Sure, yeah. So, you know, methods is in our name. Obviously, we're really interested in new methods, things that are novel and cutting edge, really advancing what's possible to learn in terms of biology. I think not everybody realizes that we have other formats that I think are equally important. So one I'd like to highlight is called analysis. So this would be a comparison of either methods or software tools. You know, these tend not to be perhaps the splashiest papers, but I think they're really important for evaluating performance of methods and for, you know, ensuring that they're reproducible, and resources as well. So we don't publish a lot of them, but again, I think, you know, collections of tools or even, you know, huge data sets, these are things that we are actually interested in publishing. And we also publish reviews, perspectives, and opinion pieces. And I think these are also important. They nicely supplement and sort of convey what's going on in the field. You know, I think opinion pieces is something that we're definitely going to be focusing a bit on this year. And I just wanted to say, you know, I feel this meeting has been a lot about the transcriptome especially, but you know, I think what we're interested also in is the other omes that are maybe not getting as much attention at the moment. So the proteome, the metabolome, the lipidome, the glycome. I think, you know, these are much harder to study than the transcriptome, you know, because of the diverse chemistries and the dynamic range issues and the temporal issues. But you know, so they're still very much in their infancy, these fields, but this is something that we're looking forward to seeing develop over the next couple of years. So I'm particularly excited about that. The single cell versions of these methods.
Mm-hmm. Definitely. And Lucia, did we get you for this question yet?
Yeah, so I mean, we are a broad journal of cell biology. So I mean, as I mentioned before, we publish these papers on cell biology, but we also have this tools format for new methodologies, for new data sets, new resources, and we are also publishing the development of these new methodologies and yeah, everything with a focus on cell biology, but we have also that more methodological part in the journal. So we try to cover both the cell biology aspect, but also these new technologies that may help advance the cell biology field for analysis or for quantification. Yeah, we try to cover both. Yeah.